Hello everyone. Today we'll be solving Cambridge International AS and A-Level Biology Paper 5 Planning, Analysis and Evaluation October-November 2021 Paper 51. Question number 1. Baker's yeast Saccharomyces viceae has two metabolic pathways for the production of ATP: aerobic respiration and fermentation. Oxygen is not used in fermentation. These pathways are summarized in figure 1.1. Aerobic respiration: glucose glycolysis produces ATP, produces pyruvate, leads to carbon dioxide, acetyl coenzyme A. Krebs cycle, oxidative phosphorylation, and then it produces carbon dioxide and water. Also produces ATP in that particular process. And then there is the process of fermentation, where glucose in the process of glycolysis is converted to pyruvate, releases ATP, and then the pyruvate produces carbon dioxide, turns into ethanol, and the ethanol is reduced into ethanol. Some students were researching details of the respiration of yeast in their research. The students found information about the Crabtree effect. This effect occurs when yeast is kept in high concentrations of glucose. The yeast gains sufficient ATP from substrate-linked phosphorylation during glycolysis. Yeast does not carry out Krebs cycle or oxidative phosphorylation and therefore does not use oxygen. The students wanted to investigate the effect of temperature on the activity of yeast and decided to use yeast solution in which the yeast obtained all of its ATP by fermentation. The students made a suspension of yeast from 1 gram of dried yeast and 25 cm³ of water. The suspension was left for 2 hours at 20 degrees Celsius. After 2 hours, 25 cm³ of glucose solution was added to the yeast suspension and the mixture was stirred. Some of the mixture was added immediately to the apparatus shown in figure 1.2. The student measured the volume of carbon dioxide produced by yeast fermentation. So, we can see the stopwatch, we can see thermometer, water in the water bath, yeast suspension and glucose solution, and we can see the gas syringe. And the question says state the independent variable and the dependent variable in this investigation. So guys, independent variable is the particular parameter that we change to get the result. Now in this investigation we are measuring the effect of temperature. So if we are measuring the effect of temperature, we are going to change the temperature, and that's how we are going to see what kind of effect that brings about. So our independent variable will be temperature. And then dependent variable is that variable that is basically our result. In this case, we can see that the volume of carbon dioxide that will be produced and collected in the gas syringe is actually our result. So dependent variable, volume of carbon dioxide; independent variable, temperature. Explain why the students stirred the mixture of yeast and glucose before adding it to the tube in apparatus in figure 1.2. So basically, when the students stirred the mixture, it ensured that the glucose and the yeast were mixed homogeneously, and it was like completely mixed. That's why the students stirred it. Part 3. Describe a method using the apparatus set up as shown in figure 1.2 that the student could use to find the temperature at which yeast carries out fermentation and its maximum rate. Describe a method using the apparatus set up as shown in figure 1.2 that the student could use to find the temperature at which yeast carries out fermentation at its maximum rate. Your method should be set out in a logical order and be detailed enough for another person to follow. You should not include details of how to make the yeast suspension or how to set up the apparatus. Alright, because in this particular experiment we are looking at the effect of different temperatures, so we must choose like a minimum of 5 different temperatures. Alright, and then we must also make sure, all right, that we are using the same volume of yeast in every experiment, same volume of glucose in every experiment, all right. And whichever temperature we are using, we must ensure that we are maintaining that particular fixed temperature throughout the experiment using a using a water bath, all right. And if we are doing the experiment, all right, at a higher temperature than that of the room temperature, then in those cases we allowed those particular glucose and the yeast mixture to equilibrate to that particular temperature before mixing those substrate and before mixing the glucose and the yeast together. And then we will also make sure that we leave those yeast and glucose mixture for a certain amount of time, example like 10 minutes or 15 minutes, all right. And then we record the volume of carbon dioxide that is produced. We can also repeat the same experiment by replacing the yeast, active yeast, with just water, and see if there is any effect of adding water to that particular glucose solution, all right. And we can also use it as a reference, or a reference point for comparison. For each temperature, at least 3 replicates of that same experiment should be done so that we can calculate the average of the volume of gas collected, all right. And then we can repeat the experiment if possible with a narrower range of temperature, all right, which seems like gives the maximum rate at the temperature at which it gives like the highest rate. We can repeat in that particular temperature again. This particular experiment is a relatively low risk experiment. However, there is a risk with for example, there is a risk with dealing with yeast. Some people can be allergic to yeast, so wearing gloves and goggles or mask is important, all right. Throughout the experiment, pH can be a factor that needs to be controlled, so using a buffer solution in all of the experiment can be also a way of maintaining the pH throughout the experiment. So, guys, the question said that describe a method using the apparatus set up as shown in figure 1.2 that the students could use to find the temperature at which yeast carried out fermentation at its maximum rate. You should set out in a logical order and it be detailed enough for another person to follow. So what the student can use is that the student can use at least a 5 different temperature, example like 20, 25, 30, 35, 40 degrees Celsius. For each experiment, the same volume of yeast glucose mixture is used in test tube. The student can use a water bath to maintain constant temperature throughout the experiment. For each temperature, the yeast suspension and the glucose solution was allowed to equilibrate to reach to each temperature. The mixture should be left for 10 minutes. After putting the mixture together, the mixture should be left for 10 minutes and the volume of the CO2 produced then can be measured. A control experiment should be done by replacing the yeast with water. For each temperature, at least 3 replicas should be done to calculate average. For maintaining constant pH, buffer solution can be used. This experiment is a relatively low risk experiment. However, if the student is allergic to yeast, gloves, goggles and mask should be used. For the part where to find out the maximum rate, all right, we can repeat the investigation with a narrower range of temperature around that which seems to give maximum rate. Now the next question, part 4. Complete the sketch graph to predict the results that you would expect from the method that you have given in part 3. Include x's with units in your answer. So the horizontal axis will be labeled temperature because we are increasing temperature. That is our independent variable. So temperature slash degree Celsius. And then the y-axis, the vertical axis, will be carbon dioxide volume slash cm cube. With temperature, what's gonna happen is that we're gonna see that there is an increase in rate of reaction, and then finally we're gonna see that it will again decrease at a certain temperature. Part B. Scientists have discovered that some other species of yeast show the Crabtree effect. The Crabtree effect means that in high concentration of glucose and with oxygen present, pyruvate is not metabolized in mitochondria. So aerobic respiration does not occur. Yeast species that are able to maintain their levels of ATP production when conditions suddenly change and no oxygen is available show the Crabtree effect. An investigation compared S. cerevisiae with the yeast Candida tropicalis to see if this other species also showed the Crabtree effect. 3 flasks containing suspension of S. cerevisiae is in a growth medium were initially kept in different conditions for growth. After a set time, the yeast in each flask was transferred to a flask containing a fresh medium with high glucose concentration and no oxygen. Experimental condition. All other conditions were standardized. The same procedure was repeated for C. tropicalis. The condition for growth and the experimental condition in the flask are summarized in table 1.1. Flask 1. Conditions for growth. High glucose concentration with no oxygen. Experimental condition. High glucose concentration with no oxygen. Flask 2. We have high glucose concentration with oxygen. And in flask 3, water, no glucose with oxygen. Alright, and in experimental condition we can get high glucose concentration with no oxygen, high glucose concentration with no oxygen for all of the experimental condition. Samples taken from the flask kept in the experimental conditions were put into the apparatus shown in figure 1.2 to measure the volume of carbon dioxide produced by the two yeast species. The results are shown in figure 1.3. The final volume of carbon dioxide produced by the sample of yeast from flask 1 at 30 minutes is shown as a maximum volume of 100% in figure 1.3. All other volumes were calculated as a proportion of this maximum. This allows a direct comparison to be made between the two species. So S. cerevisiae, we can see flask 1, flask 2 and flask 3. Percentage of maximum volume of carbon dioxide produced. Alright, for C. tropicalis, we can see flask 1, flask 2 and flask 3. Time after transfer to experimental conditions and time after transfer to experimental conditions in here. State and explain the evidence in figure 1.3 that supports the idea that C. tropicalis does not show the Crabtree effect. If we look into the data, we can see that this S. cerevisiae in the flask 2 condition, the flask 2 condition is the growing condition is high glucose concentration with oxygen, and high glucose concentration with no oxygen is the experimental condition. So when we leave high in flask 2, when we in the experimental condition, when we leave C. cerevisiae at high glucose concentration but no oxygen, we can see that it produced the same level of carbon dioxide as in that with oxygen. Whereas when it comes to C. tropicalis, the amount of carbon dioxide decreases. Since the amount of carbon dioxide decreases, this provides evidence that C. tropicalis there is a lower production of carbon dioxide. When transferred from condition with oxygen to conditions with no oxygen, yeast have to use fermentation, which is anaerobic respiration, but C. tropicalis does so at a much slower rate, as we can see. So the in order to have the Crabtree effect, so thus less CO2 is produced, because there is no Crabtree effect in here. Because the question says state and explain, so we'll get a mark for data code. The question says state and explain the evidence in figure 1.3 that supports the idea that C. tropicalis does not show the Crabtree effect. Guys, let's have one more time understanding what is a Crabtree effect. A Crabtree effect means that in high concentration of glucose and with oxygen present, pyruvate is not metabolized in a mitochondria, so aerobic respiration does not occur. All right, so how to prove that? If the if without the presence of oxygen we're gonna see the same effect, same amount of respiration, then we will understand that that particular yeast has a Crabtree effect. Now, once we see that flux 2 provides us evidence as C. tropicalis does not show Crabtree effect, as C. tropicalis produces lower carbon dioxide in anaerobic conditions. We can see when flux 2 in flux 2 with C. tropicalis we allowed it to go in anaerobic condition, we saw that the rate of respiration decreased, whereas in S. cerevisiae it did not. That means it was doing anaerobic respiration from before, so we give oxygen, we don't give oxygen, it doesn't necessarily matter for it. For flux 2, C. tropicalis has 65% versus S. cerevisiae which has 93% CO2 as of the maximum. This means that transferred from conditions with oxygen to conditions with no oxygen, yeast have to use fermentation, but C. tropicalis does not does so at a much slower rate, and there is no Crabtree effect, thus less CO2 is produced. Question number 2. Species diversity is often used as a simple way to compare the biodiversity of different regions of the world. A student read that the species diversity decreases with increasing distance from the equator. The latitude at equator is 0. Latitude increases north and south of the equator. The student selected data from a database which holds records of the annual bird count in North America. This annual count is carried out each year during one day in the winter. Volunteer counters count all the species of bird that they observe in each location. The area of each location is the same size. The student found data from 17 sample locations with Delmarva Peninsula on the east coast of the USA for 2005. Figure 2.1 shows 17 locations within the Delmarva Peninsula latitude, and we can see distance in kilometers. All right. Now, the number of species of birds and latitude. Describe the trend shown by the scatter graph in figure 2.2. All right, so in figure 2.2, when we are looking into this particular scatter graph, we see that there is no clear relationship. As the latitude increases, the bird species diversity almost remains the same. The student analyzed the data using Spearman's rank correlation test. State 3 reasons why the statistical test is a suitable way to analyze the data about the species diversity in figure 2.2. Actually, all right, basically when using Spearman's rank correlation, all right, we can allow the number of bird species and the latitude, all right, the two data points which are independent of each other to correlate. All right, the data is discrete and can be ranked. You can see over here there are a total of 17 pairs of observation over here, all right, which suggest a sort of the scatter graph suggests a sort of decrease in relationship. State the null hypothesis for this investigation. So for this investigation, the null hypothesis will be there is no correlation between the latitude and the number of bird species. Part 3. The student collected the value of Spearman's rank correlation coefficient RS as negative 0.359. Table 2.1 shows the probability table for Spearman's rank correlation test. The number of pairs of measurement 15, 16, 17, 18 and P is equals to 0.05. Now our exam probability is 0.05, and since we have the number of pairs of measurement, we have here total of 17 pairs. So at 17 pairs, our critical value is 0.485. Discuss with reference from table 2.1 the conclusions that can be made from the analysis of the data collected by the student. So the critical value is for P equals to 0.05% is equals to 0.485. So the question was discuss with reference to table 2.1 the conclusion that can be made from the analysis of the data collected by the student. We can see this RS value is negative 0.359 collected by the student, and the critical value for P 0.05 is 0.485. The value for RS is less than critical value. That means the null hypothesis is accepted, that there is no significant correlation between latitude and number of bird species. However, there is a weak negative correlation between latitude and number of bird species as shown by the negative value of the RS. C. The student was investigating the statement species diversity decreases with increasing distance from the equator. Discuss the limitations of sampling method and of the data collected in supporting this statement. Okay, the question was, discuss the limitation of the sampling method and the data collected in supporting these statements. So if we look into the way the student collected the information, the student selected data from a database which holds a record about the annual bird count in North America. This annual count is carried out each year during one day in the winter. Volunteers count all the bird species that observed in each location. The area of each location is the same size. So counting only one day, it brings about a lot of limitation. First limitation that we can think of is that uh, they have count only the bird species that they could see. Obviously they did not count the number of individuals, or how many species, how many organism of each particular individual in each particular individual species was available. The student, that's why, may have easily missed birds, those that flew away. There are no data on the latitude south of the equator, as we can see from the information. All right, we can see that there is no data from the south of the equator. All right, the student only sampled on one day in one year, which is a little bit problem with the timing in terms of sampling the data. Small birds such as the ruby-crowned kinglet, Regulus calendula, are found throughout North America. Figure 2.3 shows a ruby-crowned kinglet caught in a mist net. Mist net are used in the surveys of the bird population. Describe how the mark release recapture can be used to estimate the population size of a small bird such as the ruby-crowned kinglet. In the mark release recapture, the birds are first caught within an area, and the total captured count, they are marked and then they are released. The marking should be done in a obvious manner, like clipping wings, clipping a few of the bird feathers. All right. However, not such a way such that the bird's survival is endangered. And then maybe several days can be provided, all right, for that those birds to mix with the original population, and then catch a second sample and count the total number of birds, and from that the total number of marked birds are also counted. So guys, we are done with the question paper for today. uh, if you like the videos like this, and if you want videos like this more to be posted in the channel, please like the video, share it with your friends. All right, and I will give more videos like this. If you have any question paper in your mind related to chemistry and biology which you want me to solve, definitely post it to me, and definitely I'll help you solve it. All right guys, thank you, bye bye.